Yeah, I was going to talk about three things today that are all centered around behavior, about problems that animals have to solve in order to survive and reproduce. And I think before I get into that, I want to pick up on something that Pavel just said about the importance of studying behavior and the danger of focusing too much on neuroscience, on neural substrate, and forgetting the putting behavior at second stage. I think this is somewhat related to the three levels of understanding by David Marr. I'm not sure how many of you are familiar with that. And what is a famous neuroscientist, was it theoretical neuroscientist, and he had an imperative, a dogmatic view on what it means to understand something. Any machine carrying out an information processing task, what does it take to claim understanding of how the brain works? And he was, again, dogmatic about it, and he said you must understand and describe it at three levels. And the first level that he calls computational theory is you have to first understand what's the point in the first place. Why is the animal doing what it's doing? What is the adaptive benefit? If you don't understand that, then you're already, I think, missing out. And I think that's something that we all should keep in mind whenever we study the brain. I think it's really useful to keep track of why the animal is doing what it's doing. What is the ultimate purpose? The second level of David Marr is algorithmic. He said you must know what's the nature of the computation that's being performed. So what's the nature of the input? What sensory modalities are being used? What's the nature of the information that's coming in? What is the algorithm that's being computed? And what's the nature, the behavioral nature of the output? If you don't know that, again, I think you're already getting on thin ice. And then the third level of David Marr of understanding is the mechanistic implementation. How are these computations realized mechanistically with neurons and synapses, maybe rather than transistors in a computer chip? How do you actually realize this mechanistically? And I think I fell victim to a trap. And the trap is if we start with Marr level 3, if we do classic neuroscience and we observe neurons and synapses in action, and then we get a lot of data, something we also discussed over lunch. You can collect enormous amounts of data. You're drowning in data. You're wading knee-deep in data, and you're now trying to extract information and knowledge out of these data. And if you are not anchored, a little bit at least, in Marr levels 2 and 1, you're fucked. I think you're really royally fucked. You are now looking for correlations. You're doing dimensionality reduction. You're doing PCA and principal components, and then you get low-dimensional trajectory through a high-dimensional phase space. And if you're lucky, it's a ring that loops back on itself. But ultimately, how that helps the animal survive and reproduce is usually not clear at all. I think this is really one of the problems in modern neuroscience is that we are too obsessed and stuck with data collection, which is getting easier and easier. It's getting cheaper and cheaper. And then we have these high-dimensional data sets, and we hope that if we stare long enough at them, that inspiration will arise. And guess what? It doesn't. It gets more and more confusing. And that's where behavior really, really helps, and behavior in a way that makes sense to the animal. Not a mouse that has to drive a car through a forest of vertical stripes or has to poke left when a left whisker is being touched and then do a nose poke. Actually, behaviors that matter to the animal. If you don't, I think that's to me is the biggest challenge right now. And I think that's why I'm so happy to be here and to engage with the people here when it's about the science of intelligence. I think most people that I've talked with today realize that the challenge is to understand intelligence at the behavioral, at the level of embodiment and engagement with the world. I think Benjamin and I even coined a term that's non-neuronal neuroscience, right? Or neuronless.
Brainless neuroscience.
Brainless neuroscience, yes. Brainless neuroscience. It's doing neuroscience without looking at brains. And I'm semi-serious about this. I also told this already to Ian Cousin, is the real neuroscientists are the people who work at Marr level 1 and 2. because they actually help us refine and pose the critical questions. And then the neuroscientists who actually look at brains, I think, are just doing the control experiments for the behavioralists. I think we are sort of the bitches for Pavel and Benjamin and Ian Cousin, who actually pose they're the real hard-carrying neuroscientists. Anyway, so that's my introduction. And what I'm going to talk about today is, to a large degree, our behavioral studies in larval zebrafish. We will touch a little bit on connectomics and brain-wide imaging, because you can't really avoid it. I'm getting gang-pressed into talking about that, because otherwise people don't take me seriously. And also that's how I maybe made my name at the beginning, is to leverage the larval zebrafish, because we can do brain-wide recordings. We can look at all the neurons being active in the whole brain in an animal that's engaging the world. And that's what people find extremely attractive and appealing. And I'm just saying, beware of that. Right? I think it's not really clear how useful that really is if you don't know how to phrase the correct questions. So we will talk about 3 things today. I'll first talk about attention and attentional switching and staying focused or disengaging with a task. That's the first topic. The second, we'll talk about the role of the autonomic nervous system and the role of the body when it comes to motivational states in an animal. So that's something we've discovered recently, that what an animal will prioritize when it engages the world. It doesn't just care about what's happening in the outside world, it also cares what's happening in the body. It's also something that's a little bit neglected, is that when animals engage, it's very important what's the state of your body, state of your heart, of your gut, of your blood sugar levels. So we'll briefly touch on that, and then I will end by talking about giving up, depression, hopelessness, and the role of astroglia in this process of long-range integration. I'm hoping to, I will give myself 15 minutes for each of these, maybe 20 minutes for the first, 10 minutes for the heart, listen to the heart, and then 15 minutes for futility, hopelessness, giving up, and then I should aim to finish at around 5. Is that right? Yeah. All right, then we'll start with the first topic, and that is about an issue that when we ask animals to perform a certain task, we are often disappointed. That's not just for any animal, I would say, including human animals, including my students, my grad students, my colleagues, that we give people a certain task, and then if they don't perform optimally, we are disappointed. And that's also true for animals, that the performance index goes to 80% or 90%, and we always think you should be able to do better than that, especially if the task is simple. And the first story is about the reason why animals might not perform perfectly. And what you learn is that there are 2 reasons why they might fail, why they might disappoint. One is about competence. They just can't do any better. Okay? We understand what that means. There's a certain task, and I just don't have the competence. The second reason why I might not perform is because I don't care. I'm not focused, I'm not engaged. I'm disengaging with the task. I choose not to engage. I could if I wanted to, but I'm just distracted. And I think those are 2 very different reasons for underperformance. You can probably all relate. I can come up with countless examples, but I think that's not necessary. And the first story is about how we realize that this is even happening in a larval zebrafish. And the first story is about how we can separate those 2 modes of failure, competence versus focus, and how we can then even use this to predict and find substrates or find reasons for failing in one or the other dimension. And there we can briefly dive into nature versus nurture. Is it they are genetically predisposed for failing in one or the other, or is it the environment? So that is going to be the first part of the talk, and we'll get into that right now. Are we ready?
Yes.
Yes. Okay, so this is, I think, is an introduction about the second part. So we'll, okay, to the larval zebrafish, system for understanding interactions between the brain and the body. We can do brain-wide activity measurements, and you should be already a little wary about that because I told you that this is mostly distracting and will lead you astray. We can do behavioral readouts in a larval zebrafish, and we can also look now at internal organ dynamics, looking at heart rate and so forth. It's just gives you access to everything you want. The behavior that we'll study is the optomotor response. That's the task. So this is the task that our fish will have to perform in. And the performance means, in this case, just hold your position in a moving stream. It's not that trivial. Huh? You're being swept away by a stream, and your task is just hold your position or minimize retinal slip. That's the task. And in the wild, it's going to look like that. We have a larval zebrafish that is being dragged backwards by a moving stream. And you can see that's an animation. That's not a real movie, but it's pretty good animation. Christian Herrera made that. And how we mimic that in the lab is that we have him swim in an arena, and we're just moving now the world forward or sideways, and we're asking the fish to swim along. This should be relatively clear that either being swept backwards by the water column or seeing the bottom move forward are identical from the point of view of the animal. Those two scenarios is just a switch in reference frame, but for the animal, that's exactly the same problem to solve. Is you just need to follow the stripes or the patterns that are being projected onto the bottom, and if you do that, you hold your position in a moving stream. To make it a little bit more realistic and challenging, we give it a noisy stimulus. You can see only a subset of the dots are moving, so it's a noisy river bottom that's moving past the fish, and he's asked now to swim in the direction of the coherent dot motion. That's a useful stimulus because it's mathematically clean at least, and we can dial in the amount of noise linearly and cleanly by just changing the percentage of dots that move coherently, whereas all the other dots move randomly. That should be relatively clear, right? We can now dial in the coherence level from 0% coherence to 100% coherence, and that should make it easier and easier for the animal to perform well in the task. And the other nice thing about this, so here on the left you just see fish swimming around randomly. I just put that in there for people who never seen larval zebrafish. That's what they do normally. If there's no moving dots, they just swim around. And you get colorful trails that tell you where they're swimming. This is sort of a background, baseline swim behavior. But here's the task, is where they need to follow this motion. And so the trial usually is we start with random coherent dots where the fish doesn't have anything to do. And then for 30 seconds, we present dot motion that always stays orthogonal to the body axis of the fish. So we don't make it that easy. It's not just that we move the dots forward and he has to swim forward. We move the dots orthogonally, and the fish ever has to turn and align himself with the dot and swim along. But the moment he does that, the dots will move along. Yeah? We are mean, actually. We are moving, we are locking the dot motion always to 90 degrees to the animal, such that now what this fish will do, it will just string along one choice after the other, where it either has to go right or left to follow this motion, so he can make a correct choice in going with the dots, or he can make an incorrect choice in going in the opposite direction. And we can do this basically until the cows come home. So this is nice because fish don't habituate, don't desensitize. They will do this effort tirelessly. Which also makes sense because if they're out in a moving stream and they don't want to be swept away, they have to keep doing that as long as the background is moving to avoid being separated from the group and being swept away. So it allows you to collect a lot of individual decisions. Every swim bout here, when the dots are moving, is a decision. Where we can score. Was it correct or incorrect? So that allows us now to acquire a performance index. These are the people who did all the work. It's Kumaresh Krishnan, who just finished his PhD, and Adrian Fairhall, who co-supervised this project from Seattle. Yeah?
Just for clarification, in real life there would be water and a stream and pressure, right? But it's in this case there's nothing for the animal.
If the water motion is homogeneous and laminar.
So it wouldn't really have a high pressure on the...
There's no pressure.
Yeah, okay.
So if the reference frame of the river, the moving river, is the reference frame of the fish, then there is no change in pressure, flow, or anything. Right?
Okay.
This is like relativity, Einstein, right? I think this is a reference frame problem. So the only thing, unless the fish is getting close in the river, in the real river, so if you want to study rheotaxis, then the only time the animal can actually know something about the water movement is when they're getting close to the wall or the bottom, where you get a gradient of flow velocity. And that's a separate story. We know that they can pick up on these flow velocity gradients, can calculate the curl, the resulting curl, and use that to navigate water flow, even in complete darkness. But for that, they have to be close to the wall. The moment they are away from the wall, you are in a homogeneous reference frame condition where it's a Galileo transformation and there is no change in pressure or flow. But it's
That's unless they're orthogonal to the flow of the water. No? So,
no, I mean, imagine you are, briefly, if you put a tank, aquarium, in a train, and you run the train at a constant velocity. Do you think it matters in the tank, in the train, which way the fish is pointing? No, clearly not, right? Or if we put a tank here, and we know that the Earth rotates eastward at 1,000 miles an hour, that velocity is completely irrelevant. So the absolute motion of the water column is, it's again, this is the Galileo transfer. Is Galileo already realized that? And modern scientists have forgotten about this. So this intuition that we think that if we are in a moving water column, somehow this must interfere, is a wrong intuition that we share with almost everyone, but it is really, really wrong.
Sorry again for...
Yeah.
Shouldn't you feel acceleration in the moment where the direction changes?
But only self-induced, right? So if you do that in a lake, if you're swimming around in a lake, you feel acceleration every time you're moving. Okay? That's trivial acceleration. If you're in a river, and if you're moving at constant velocity, the moment you're now changing your local velocity, you see the same, you experience the same acceleration in a river as you do in a lake. That's the short answer. Because when you're turning your broadside, you're not slowing down, right? You're moving in the reference frame of this river, right? I mean, think about it just being in a train, having a tank in a train, or if that's not natural, think of it, I think the example I used is a long swimming pool, on a ship that's going fast in one direction, right? You have a swimming pool lane, right? And the ship is going in that direction. So this is a water column that's moving 20 miles an hour. It's a river.
And the thing what confused me about that is that the ship would suddenly change direction. And in that brief moment...
But the river doesn't change direction, right?
But in this experiment, the dots suddenly change direction.
Oh. So the question is, I mean, if the dots change direction, that's true. I think the reason why we have the dots change direction, that's a little bit, you're right, actually. That would be like the fish being in an eddy or in a whirlpool, and then there should be centrifugal forces and all of that. That is true. So the changing of direction is something we just do to collect more data. But you're right. In a real world, if the river would change direction, if it would slow down or accelerate, the fish should be able to feel that with his vestibular organ. And they usually, so that's a good point. I mean, this is actually not bad to discuss this in detail because that assay is at the core of the whole story that I'm going to tell you. And it's good that we're all on the same page. What the fish is trying to do, exactly what's the assay, from the point of the view of the fish, what this feels like. But in a way, what these animals do, they see the world moving, and then they try to follow. And they do this reflexively and innately, and it makes them feel good, and they like doing it. And we just want to know how well are they performing. And the preference index that we'll see a lot is if you make the correct decision, you get a 1. If you make the incorrect decision, you get a -1, and then we just average. And then we get a performance index that can be 100% or -100%, and Bob's your uncle. So this is what we do. And that's the assay. And now we can do this a lot. So it turns out, because it's a stochastic behavior, we need a lot of data. And the way we did that is not just have one of these dishes that you just saw, but we have here 8 on a, we can scale this up, we make it high throughput behavior, and we can make it even more high throughput. And now we have 16 units times 4, no, 8, yes, no. 4 times 4 is, no, it's 8, whatever, it's a lot, right? 16 times 4 is 96, I think, and then we can have each, and so 64 fish, each does 30 trials in 90 minutes, 150 seconds baseline, where they just swim around, 30 seconds test, where you have to now pass your exam, where you have to make right or left decisions, which is hard because it's noisy. So we get, in 90 minutes, we get 1,920 trials. And each trial lasts 30 seconds, so we can collect probably up to 20 or 30 decision points in each trial, which allows us to assign a performance in each trial. And now what we find, so here is the collected data. On the x-axis, the turning angle, which can either go in the direction of the dots right here, or it can go here to the left when the dots are going in the other direction. Baseline condition is shown here as a dotted line, it's symmetric. They make sometimes randomly large angle choices, but generally speaking, the behavior looks like that. Sometimes they go forward, sometimes they go left, sometimes right, but there's a bias. And if we calculate the performance at 100% coherence, it's about 70%. That's really disappointing. We're very disappointed in our fish. It's not that hard, right? I mean, it's not that difficult. This is the 100% coherence level. They should be able to do better, they don't. So we're very disappointed, and before blaming the fish and calling them incompetent, we now wanted to look really at the structure of the data. And in order to do that, we first looked a little bit, can we predict when they would fail? What's the failure mode? Is there anything that allows us to look at this fish and say, why are you not getting this? So one thing we looked at is at the baseline bout rate. You remember there's 150 seconds baseline where they're preparing, they're just swimming around. And what we found is that if the baseline bout rate, so usually it's about 60 bouts per minute, so one every second. They make a decision every second, but at the beginning, no, here's baseline bout rate. Those are the fast fish, the hasty fish that swim a lot during the baseline, and these are the slow, the sluggish fish that don't swim that much. And what we see already is that the performance is somewhat correlated with the baseline bout rate, which means that the slow fish do better. So if you take your time already during the baseline, then you're predicting that you do better than if you are swimming a lot. And that might even intuitively make a little bit sense. That gets into competence, is if you don't take your time when you make decisions, you're more likely to make mistakes. It's the time accuracy trade-off. So there's already some correlation. We model all of this with a drift diffusion to bound model, classically, that allows us to model this decision process. And during the baseline, we can plot now the baseline bout rate against the performance score. And what I'm plotting here is just during baseline, where the performance goes from -1 to 1. This just means that the fish sometimes, this is arbitrary, right? There is no actually correct or wrong decision. We just assign randomly a correct choice, and then we see that the behavior it has a Gaussian distribution. There's nothing special here, right? Our model and our real data just tell us that the distribution in which fish turn has a Gaussian distribution, and it doesn't really correlate with the baseline bout rate. That's more like the baseline control experiment, and this was for me an epiphany. When I saw this for the first time, I had a moment where I suddenly looked at that and I said, wow, there's something that makes sense. So first of all, our model, our drift diffusion model, just predicted that the fish would perform at a level of about 0.7 here. Sometimes they get it wrong, sometimes they get it right, and that's because of incompetence. They are just not very good at getting the 100% that we expect. When we saw that, I at least was thinking, so again, here is performance index. This is chance level. This is perfect performance, 1. This is always going in the wrong direction. And I would argue what you see here is 2 things that are really interesting and that look different from that. And what you see first is that there is a high performance cloud. This is when they're engaged and actually doing a really good job. And here you see even this cone-shaped, that the slow fish perform really well, and the hasty fish during baseline tend to sometimes get it wrong. Yes?
Do you have reaction time of the fish?
Yeah, it's the same. So the reaction time is usually fast and uniform. It's 100 milliseconds, 100 to 200 milliseconds, sometimes a second after the onset of the coherent dot motion.
I was wondering whether the speed of the decision is anti-correlated to the accuracy.
That's correct, yeah. So the longer they wait, the better they perform.
Because that is not in this graph, you are looking at the baseline.
Right, no. So there is a preceding story where we just look at integration, accumulation of evidence, and there we find that the longer the fish waits, the higher the performance index. So this is a linear integrator. We are having a leaky drift diffusion to bound model that predicts that the longer the fish waits, the better they will do. That's almost like a trivial integration component. This is a little bit more subtle because, as you've pointed out, this is the baseline bout rate. This is more like looking at the baseline behavior and predicting how they're going to perform in the future, and that tells you more a little bit about the internal state of the animal rather than about the online performance. But that's a good point. Yeah.
I also have a clarification question.
Yeah.
I'm sorry to interrupt. How do you tell fish make a decision?
They swim.
They swim.
Make a bout. So these fish, I have to. So maybe I haven't said this. This was in the movie. Fish, larval zebrafish, swim in bouts, where they wait a second, then they make 100 millisecond bout, and then they wait another second. And so every bout is a decision which is preceded by about a 1 second integration period. Good, thank you. That is important. All right. Now, one thing is this high performance cloud. Before we had these are 2,000 data points, 2,000 trials, 1,920 trials. Before we had that, we only had a few scattered data points, and there we had sort of a few examples here that we called the dumb fish. But now, if you look at the distribution of this cloud, this does not look like incompetence. This looks like negligence. This looks like they're ignoring completely the stimulus. The shape of this cloud is the same as this one. It's not that they are sometimes going in the wrong direction. They are going in the wrong direction with equal probability than going in the right direction. These fish are ignoring the stimulus. They're not paying attention. They are just not attentive at all. So when I saw this, I said there are 2 states here. There's the engaged state and the disengaged state. And the way how we can separate those 2, I'll talk about it in a moment to convince you that this indeed is a Gaussian that's centered around chance, and this is a gamma function that is centered around that's squished against the ceiling.
What is one data point here? So how many decisions is that? Is that one fish, or is it one certain duration in a given fish?
Thank you. One data point is 30 seconds of continuous decision making. It's about, on average, 30 data points. Minus ones or ones, go into one data point. It's 30 seconds of one trial.
Okay.
And then the average decision frequency is 1 hertz, so you get about 30 decisions for one data point. Sometimes, for those guys, it's much fewer, right? This is where a fish is making only 1 or 2 decisions, and then they can happen to be both go in the wrong direction, and then you get a -1. Very good. All right? Yes?
Are there fish who only do like the right choice and some who only do the wrong choice, and then fish who do both?
No, I think it's basically described here. This is a trial. And you see there's even the ones here are distributed over several fish, right? So the distribution here also happens to be representative for individual fish. This is 64 fish that added their trials to here, but the individual fish do not deviate roughly from this distribution. We'll talk about individual differences now between strains in a moment, right? How we can leverage this now to ask the reason for failure. So again, I think this is the main point here. These are inattentive fish. They're not paying attention. And these are attentive fish that are performing really well. So we can quantify now for an individual animal or a group of fish, how often do you ignore the stimulus? But if you're engaged, how well are you doing? How sharp is this high performance cone up here? So we can quantify these two elements independently for any given animal. And how exactly we do that is shown here. So we integrate here over this distribution, we get a Gaussian, we fit this one with a Gaussian, and we fit this high performance cloud with an independent gamma function. And the Gaussian just simply tells us, and first of all, we can fit this really well over all the experiments we did with a Gaussian that's centered around chance. And then the relative integral between the Gaussian and the gamma, the ratio between the number of data points, tells us how often that are these fish inattentive. And so what's the percentage? In this case, I think it's the, I think we'll have to get to that. It's roughly, I think this is, they are paying attention about 70% of the time, and 30% they're inattentive. But if they're attentive, how well are they doing? And that means the closer they are to 100%, the better is the competence. So we can separate this into focus is the relative contribution of the Gaussian, and if they are performing, how well are they doing? That's a level for competence. And yeah?
I'm confused. So one data point is a fish or a time point for a fish?
One data point is a 30-second trial of a given fish, and each fish is going through 92 trials. So I think there were 94 trials for each fish, and we have 64 fish.
So you set this entire experiment up just to do one 90-minute recording, but everything in parallel.
But then we repeat this many times, right? With different groups of fish, and I'll get to that. So this is one group of fish. And now we want to know, can we change either focus or incompetence when we change parameters, when we use different strains, when we perturb the animal? Can we now switch this, can we make them do better or worse by perturbations? That's why we have so many. Otherwise, we wouldn't need these fleet of experimental setups. Okay, so this is just the normal Gaussian distribution that's in yellow. So if yellow is big, that means the fish is not paying attention. Blue, it's paying attention. That's the W here. This is our focus attentive quantification. So if W is large, fish is attentive. If W is small, it's not paying any attention. Kappa is the parameter for the gamma function that tells us how competent the fish is. Kappa can in principle go to infinity, meaning that this high performance cloud can get like a delta function, can get, so this is open-ended, but it gives us a level for the competence. And that's important, that is independent, is orthogonal to focus. So those two parameters can be adjusted completely independent of each other. Focus and competence are completely separate domains that both can change your performance. And the final two slides is where we now ask who can we make responsible? Whose fault it is for either incompetence or lack of focus? Shall we blame the parents or shall we blame the upbringing? That's one of the questions. Or can we make the fish more competent or less competent, more focused or less focused? Is this clear, right? I mean, how we get yellow means not focused, meaning it's all in the Gaussian. If blue is high, it's all by the gamma, and the kappa tells you how well they're doing. Yeah.
Just thinking about temporal correlations, because you do these 30 trials or 20 trials or so in these 20 seconds. Now, somehow in this model, you're assuming that these are all either all not
correct
or all competent.
That's correct.
And I'm wondering if you wouldn't get a wild mixture of these things normally if they would switch at random times.
Yeah, I think we have some indication that the state of being either engaged or disengaged can last for a few minutes. Huh?
Okay, then
but I think, and we tried to do a hidden Markov where we assume that the state switching can happen within a trial, but because these are binary choices, minus or ones, it gets a little difficult. But I think yes, so that's true. That's a bit of a trick we are using here. But we believe now that the switching time constant is significantly longer than 30 seconds. That this lasts probably a few minutes. But it's a good point. All right. How can we change that? The behavioral parameters. Well, first of all, we can model that relatively simply in our state switching. We just have an engaged state and a disengaged state, and we have a switching variable that we call P, or 1 minus P, that just allows the animal to switch between those two states, and that really captures the data sufficiently well. So it's just a stochastic element that makes the fish either switch into the engaged or disengaged state. So if P is close to 1, the fish is always engaged. If P is close to 0, it's always in the disengaged state. And the question is this P, how does this vary with perturbations? I think we'll skip over that. And now we go, well, first of all, no. So here is the critical experiment. It is about nature versus nurture. What we leveraged here is the fact that we had 2 facilities run by 2 different PIs. The A facility is the one raised by Florian Engert under the control of Florian Engert. The B facility is the facility under the control of Mark Fishman, my very good friend and colleague. And what you can see already is that the fish in A have high focus and high competence, and the fish in B have low focus and low competence. That's convenient, because it means clearly that I'm a better controller of fish facilities, but it also allows us now to do an experiment where we can shift the fish. We can take the Florian fish and raise them in the B facility, and we can do the same with the Fishman fish, right? We can switch the parental contribution and the environmental contribution. And the first thing we did is we took the fish from the competent and focused fish, and we raised them in the Fishman facility. And you see the focus doesn't change, but the competence drops. This suggests already that your competence is based on the environment, on your upbringing, on your schooling, but the ability to focus seems to be just depending on the genes. If that's true, then we would expect that if we bring the bonefish from the Fishman facility over, that focus shouldn't change much, but competence, if anything, should go up a little bit. And again, this is not as clean. Certainly the competence is going up. In the focus, we also have a little bit of an improvement, so it might not be as clear-cut as that, but this suggests already that the focus is something that is innate, that you can't do much about it, but your skill or your competence might be something that is subject to environmental perturbations or contexts. And I'll just tell you how this actually really makes sense when we do explicit perturbations to these animals before we test them. And what you see here is 1, 2, 3, 4, 5 different perturbations. 2 are genetic mutants. Here we sleep deprive them, here we subject them to looms, so we just want to perturb them. And here it's more like a baseline test where we make it just harder, where we make the task harder, we go from 100% to 50% coherence. So maybe we should start with that. And here's the control. Here is during baseline. Here is the Here is the control fish that are not perturbed. Here are the experimental fish. And what you see is that if you make it harder for the fish, then the focus doesn't really change, but the competence drops as expected. It's just difficult. At 50% coherence, it's harder to do it, so your performance drops, but the engagement doesn't really change. One thing I want to point out, that all of these animals that we tested here come from different parents. There are different parental strains. For the mutants, this is obvious, they have to be. For these just happened to be different colonies, different cultures. So there's a huge variance in focus across the different perturbations, which makes sense. That's what we think. This is a genetic component that determines the focus. But the experiment itself, the perturbation, did not change this. So whenever we do an environmental, or even if we test a mutant against a wild type, we see this does not have any changes in focus, but what we see in the competence level, that any of these perturbations increase the competence. If we scare the fish in the baseline, they perform better. If we sleep deprive them, they perform slightly better. The competence is going up. Our mutants,
No, I'm just, that's what I'm doing, I'm saying.
Our mutants also, if anything, show an increase in performance. And the increase in performance, even in the mutants, is based on competence and not based on focus. So focus is something that stays relatively constant under these perturbations. And to me, that is an interesting insight, not just for baby zebrafish, but for all animals, that if you have performance problems, if it's competence, you can work on it. You can practice, you can try to get better. But if you just happen to get disengaged again and again, then there is not much you can do about it. It's best just to live with it. And just go with the flow, roll with the punches, don't try to fight that. That's maybe even more important when you try to teach students or children. If they stop losing focus and stop not paying attention anymore, there is really not much you can do rather than relax, step back, and let it play out. How about that? All right. Maybe briefly. Okay, let's continue. Second story is about what is it that might make them switch. So we find that these fish occasionally switch, stop paying attention. And the second story is this might have something to do with the internal state of the body. So maybe it's the heart rate that's going up. Maybe it's something that's changing in your body that's making you go into a disengaged state. And for that part, I want to introduce the autonomic nervous system, which is also understudied. It's an orphan in neuroscience. You all have it. It's highly conserved across all vertebrates. I think it's basically the same. It works the same way in you as it works in me, as it works in a larval zebrafish. And so here's a brief introduction. We have the sympathetic neurons. They are the ones that sit in ganglia close to the spinal cord in the hindbrain, also in your brain. And if they get active, they energize. They drive everything up, heart rate, energetic state. We have the vagal, the parasympathetic nervous system. Vagus is the most famous. Again, it's something that when activated will calm you down, rest, digest, rather than fight or flight. And this is about control, top-down control. And we also get information back. The body can talk back to the brain, and this is not through the sympathetics, that's through the vagus. All right. This is the complete autonomic nervous system in larval zebrafish, but it looks exactly the same in mammals and in humans. And we can image that, right? So we can, in this case, we can watch everything in the body and we can ask how this information is going back to the brain. And here we're going to talk about just about the heart. We talk about heart rate, and we want to know what changes in heart rate, what consequences that heart rate changes have for the emotional state of the animal. It's about the heart and the mind, and it's about the heart speaking to the brain. And what you learn is that if you listen to your heart, then you can actually learn a lot about yourself. Huh? In a way, that is one of the second take-home messages, that what the brain is doing, it's not just listening to the eyes and the ears and to the nose and the touch, it's also listening what your heart is doing. And here is a heart rate measurement, which you can easily do. We have tail curvature here. And one thing we found first is that if we introduce threads to the larval zebrafish, these are dark flashes here, and in red are salt pulses when the salinity goes up. And what you see that sometimes there's behavior, but in all of these cases, we get a change in heart rate. So the heart actually monitors, probably through the thalamus, through direct subcortical low-level innervation. The heart monitors the environment, and it will respond with autonomic changes to threats. There's also spontaneous fluctuations in heart rate, which I think are also really informative for the internal state of the animal. And here's our first, really, I think, fundamental finding. This is not easy to see. It's in pastel, so I think I'm going to show it to you at higher contrast here. In red, we have the heart rate. This is spontaneous heart rate fluctuations of the fish. In green and purple, we have OMR trials where the fish is either engaged or disengaged. Remember my previous talk, sometimes the fish just does not respond to the OMR. These are the purple trials. Huh? I mean, this is head fixed, where the fish just refuses to engage altogether. In green, the fish is responding. And what you probably can see easily here, that disengagement correlates with increases in heart rate. So one thing that might make the fish disengage is the heart rate going up. Yes?
But it couldn't be other way around.
Sure.
Fishes.
You don't know about causality yet. We'll get there. So what if we could drive the heart independently of anything else, right? What if we could put an optopacer in the heart and just drive the heart rate up? If we only could do that, no? That would probably answer your question. But we'll... First of all, what we convinced ourselves is that there is the OMR probability, the probability of engagement versus heart rate has a very, very strong relationship. So if heart rate goes up to 3 hertz, I mean, there seems to be a really strong, again, the causality is still not clear, but it's very, very strong. And this is the latency in which the fish responds to the OMR. So if they wait long, then the heart rate goes up. It seems that the behavior, the engagement with the world, seems to be highly, highly correlated with the heart rate. And now, yes?
I have a question to these slides. Yeah, to this one.
This one?
Yeah, this one.
Yeah.
So I know from other fish, like the Atlantic cod, that the heart rate drops when there's stress. So I wonder if you see that as well in the beginning, that there's an initial drop. And
there is. You'll see. Yeah? Okay. No, we'll get to that when we look at the details. So at the first couple of seconds, when a stress appears, you get brachycardia. Heart rate drops, and then over the course, I mean, this is minutes here, right? So initially we see a drop in heart rate, and then it starts to rise. And it's probably the vagus and the sympathetics that act in concert to control the heart rate down. Cannon-Bard versus James Lange. There are 2 theories, and I think that's what Valerie was alluding to. Is the heart rate going up because the fish is scared? And then both the behavior and the heart rate just respond to the scary stimulus? Or does the scary stimulus just drive the heart rate up and then the heart talks to the rest of the brain? So Cannon-Bard, James Lange, William James and Martin Lange propose that the heart is the primary detector, gets a direct line from the thalamus, and then communicates that to the brain. So I'm actually not scared because there's a bear approaching. I'm scared because I'm running, because my heart rate is going up. And so here is the Cannon-Bard theory. I'm scared of the bear. Thalamus gets to an emotional center behavior and in parallel autonomic control of the heart. And James Lange is that the heart responds subliminally to the bear, and the emotion centers are just responding to the heart. And we can test this now. We can test this by just driving the heart independent of any external input and asking, does that behavior change? So here, first of all, we can put an optogenetic probe in the heart, in the myocardiocytes, and then we can change the heart rate by dialing in a blue pulse of light, and we can just change the heart rate of the animal, dialing it in. And now we ask, does that actually change the behavior? And boy, does it. I think, I'm not sure why Christian Herrera, who did all of that, didn't use the same as before. So whenever the heart rate goes up, you see that the behavior drops. And this is what you see here. So at the moment we start pacing, this is a control where there is no channel rhodopsin in the heart muscles. If we put channel rhodopsin in and we clamp the heart at 2 Hertz, OMR probability goes down a little bit, doesn't like to be clamped. If we drop it even more, we get this significant drop in probability. If we go from 2 Hertz to 2.5 Hertz, the fish will respond behaviorally. And the only thing we are doing here is changing the heart rate. And the animal listens to its heart and changes its internal state, its internal motivational state, without anything else. Yes?
At what point do they run into anoxia?
Oh, never.
Never.
7 days, that works by diffusion. They will survive without any heartbeat. 7-day-old larvae. So this is not yet. And the difference between 2 Hertz and 2.5 Hertz is minimal. Now, finally, we are going to spend another 5 minutes and talk about how the connection between the heart, who's reading out the heart rate, and how is that information communicated to the motivational centers in the brain. So who's listening to the heart in the brain, and how is this information then communicated to the nuclei and centers in the brain that are responsible for executing the OMR. First of all, we found the nodose ganglion, which again you have too. That is the primary afferent nucleus for the vagus nerve that collects all of the information from the body and sends it onwards to the brain. And there we have, not surprisingly, heart rate related neural activity that tracks heart rate very precisely. So the nodose ganglion is one of the potential sensors for heart rate that broadcasts this information to the brain. And if we trace now those neurons using connectomics, what we find that one of the targets in the node that heart rate is communicated to are the astroglia in the hindbrain, are glia cells. And for that, if I may take another 5 minutes, I'll tell you another little excursion about giving up, about experiencing futility, about experiencing something that's extremely frustrating, namely when you're trying to do something and it doesn't work. In the case of a larval zebrafish, it would be if I'm trying to swim and the world doesn't flow backwards. If I'm trying to do the OMR, but it has no effect. And the person who really went to the bottom and analyzed everything and led that whole project was my very good friend and colleague Misha Arendt, so it's his story. And the way he does it, he has a head-fixed fish. We're doing the OMR now here, and whenever the fish is swimming, we are allowing him to be successful. This is a successful fish. Whenever he's swimming, the stripes are moving backwards. That's good. We can make it bad if whenever he swims, nothing moves. They keep moving forward. That's frustrating. That's futility. That's the futility tracking system, short FTS. And that's my acronym, FTS, because that's what the fish thinks if things are not working consistently. FTS is short for fuck this shit, right? I'm giving up. I'm throwing in the towel. Futility tracking system. I think it's a good acronym. And what Misha found is that first you're in closed loop, everything's good. Then you go to open loop, nothing's working. The fish is now struggling and trying, and here, yellow, FTS. Fuck this shit. I'm not doing anything anymore. And this can last for many, many seconds. The fish goes into a state of learned helplessness where he just shuts down everything. And what I'm telling you in the last 2 to 3 minutes is how this actually works. Yeah?
What would happen if it would have the opposite effect?
Then they tune down. So if they swim if too much moves, so we've done this too. Yeah? If the fish swims and suddenly the world moves too much, the fish registers, he measures an error signal. In this case, it's a good error. It's too much of a good thing. And then what they do, they tune down their efforts a little bit. They can carefully calibrate down their swim vigor in order to match now the expected speed again. So they have a feedback. They have a forward model. They can feed back the expected. And if they have a mismatch calculation machinery that detects the visual flow and compares that to a prediction generated by my motor action. And if that error is high, they will use it first to correct it. This is what the fish is trying to do here. And here's a big negative error. I'm moving my tail and it's not moving very much, and then he's trying to amp up the gain and swim a lot. And eventually, there's a threshold where you say, this is crossing a line, I'm not dealing with this shit anymore, I'm going to shut everything down. This requires an integration over minutes. So there's an error signal here in pink. There's an error accumulation where the fish is accumulating now errors. And then if that error signal reaches a threshold, it shuts everything down. And this error signal is adrenaline or noradrenaline in the medulla oblongata. So norepinephrine in the medulla oblongata is Nemo. In the fish, that's cool, that's cute. It's the medulla oblongata in fish that houses these norepinephrine neurons. And they are the ones that report the error signal, the frustration signal. And the glia cells are the ones that integrate. That's what Misha found. And I'm showing you one movie now. So this is a fish that's swimming in open loop. There's this whole brain imaging here. This is astroglia imaging here. And now everything is dandy. Everything is hunky-dory. And eventually we will switch to open loop, where suddenly here's open loop. This is frustration. This is anger. This is a pissed off fish. And here you see first huge frustration signals. And here are the glia cells slowly ramping up. And here's another engagement. The neurons light. And you see the glia ramping up. And when the glia reach a plateau, and this is a massive glial-wide signal, and these glia cells are integrating over these frustration signals. And why this is so exciting is because neurons are usually really bad at integrating over seconds and minutes. To maintain a memory of past frustration is hard to do in neurons. They would have to have some synaptic changes or phosphorylation states. Neurons suck at maintaining long time constants. Glia can do this effortlessly. So what Misha found really is that this memory of past frustration, this accumulation, this integration is not happening in neurons, which would suck at that. They redelegate this task of accumulating error signals. The neurons redelegate it to glia, who only have to keep track of one variable, that's past frustration, and they just ramp up. They have these long memory time constants. They will naturally integrate the signal, and if it reaches a threshold, now we know what they do. They release ATP, which gets broken down to adenosine. The adenosine now binds to metabotropic GABA receptors, which drive inhibitory neurons, which shut down the motor system. So this is the whole pathway. Yeah.
So the things that you just said, is that something that you know causally?
Yes.
Are involved, so you can actually.
This has been what I'm showing here. This has been causally tested with optogenetics, ablation, and stimulation. So when they know these Nemo cells, these norepinephrine cells, report frustration signals acutely. Yeah. We know the glia cells integrate, and we know if the glia cells reach a threshold, they drive these GABAergic inhibitory neurons, and if they get activated, they shut down the fish. All of this we know. This we know. That's a circuit that has been tested element by element in a zebrafish, and that really explains how fish give up when they experience a lot of frustrations. Well, we don't really know exactly how the error signal here is calculated, but we know by now it's probably serotonergic activity in the dorsal raphe, where visual information is integrated with motor efference copies, and the fish is subtracting those two. And if one is bigger than the other, you get a serotonergic signal that reports about a positive or a negative error. And then these adrenergic, these norepinephrine cells, they are the one that report frustration, pissed off, right? It's a frustration event, another frustration event, another frustration event. And the integration, the accumulation of these frustration events, that's happening in these astroglia. They are keeping track of how pissed off you should be, how frustrated. And they transfer this frustration in a system, the frustration transfer system. They transfer this to these inhibitory neurons that then can shut down the And the final slide I'm going to show you is, if that's all true, then these astroglia would really sit in a very useful position to track heart rate. Are they actually the ones that sit at this intersection between heart rate that goes up and the giving up of the animal, or the disengagement with the animal? And so we asked, are they correlated? Is astroglia activity correlated with heart rate? And boy, is this correlated. So if this is, we don't know causally yet, whether driving heart rate also drives up astroglia, but certainly astroglia match the internal state of this animal, of being frustrated, of being pissed off. There seems to be an uncanny correlation. And with that, I think I'll stop. Apologize for going slightly over time, and I'm happy to take any questions.